So again, hello and welcome. I am delighted to welcome you all to this month's Society for Marine Mammalogy Editor Select webinar, where we feature the latest exciting research that's being published in the Marine Mammal Science journal. I'm going to be standing in as host today since Editor-in-Chief Daryl can't make it. My name is Teresa. I'm a PhD student at the University of California, Santa Cruz, and one of the Society's student members at large. So it's my pleasure to welcome Michelle Greenfield. Michelle is a third-year veterinary student at the Cornell University College of Veterinary Medicine. She has a bachelor's degree from Princeton University, where she began her research with the Chicago Zoological Society's Sarasota Dolphin Research Program. On top of all her research and clinical work, Michelle is also the producer and host of the Aquedox podcast, which is a top 50 life sciences podcast and the leading podcast on aquatic veterinary medicine. So we'll definitely ask her some questions about that at the end. First, though, Michelle is going to be presenting on her recent paper with her talk titled Friends Through Thick and Thin, How Injuries Disrupt Bottlenose Dolphin Associations. A reminder that if you have questions during the seminar, there's a Q&A function at the bottom of the screen. So we ask you to post your questions there, not in the chat box. And otherwise, Michelle, it's all you.
Awesome. Thank you so much, Teresa. Hi, everyone. So nice to be here. I'm going to go ahead and share my screen. And can you guys see it? Teresa, can I just get a confirmation there?
Yep, looks great.
Excellent. All right, let me move this out of the way. Perfect. Well, like I said, hi, everyone. Thank you so much for being here today. I'm really excited to talk with you all. Though it's virtual and I can't see any of you, know that I wish I could. And we're going to talk about some dolphins today and their associations. So I'd like to thank my co-authors, Dr. Daniel Rubenstein at Princeton, Dr. Katie McHugh, and Dr. Randy Wells at the Chicago Zoological Society's Sarasota Dolphin Research Program. This project was actually my undergraduate senior thesis research, and all three of them were my advisors for this project and have been helping me over the past 2 years from original thesis and doing research to eventual publication. So it's been a great project with them, and hopefully you all learned something and we have a fun time here. So we'll get started. Let's make sure I can get to the next slide. Perfect. This is just a little fun thing. Teresa, who introduced me today, was actually my intern mate when I was working with the Sarasota Dolphin Research Program. And we found a coconut one day when we were out on the boat, and this was our coconut. So I just thought it would be fun to share. And she's in a wonderful PhD program right now. So it's amazing where everyone's going to and also like how time flies. We're also going to be using a thing called Poll Everywhere, which some of you might be familiar with, some of you might not be. Essentially, I can't see any of you, so it's a way that we get to interact a little bit here. So if you see the URL at the bottom of the slide here, pollev.com/mgreenfield, I know you're on some sort of electronic device right now, so go ahead and plug in that URL. There should be some questions that pop up as we're going. It doesn't really track you, so don't worry about that. It's just a way for us to interact as we're giving this presentation and get a chance for me to know who you are, and you get to interact and learn some stuff. So that URL will keep reappearing if you didn't catch it this time, but it's just pollev.com/mgreenfield. Great. So this is our plan for today. I'm going to give you a little bit of introduction about who I am, what we're doing, talk a little bit more about dolphin associations, then go specifically into this research project with the methods, results, and then our final conclusions. So a bit more about me. Like Teresa mentioned, I am a third-year veterinary student at Cornell University, and I'm specializing in aquatic medicine, so not your traditional veterinarian. Most of my background is in marine mammal behavior, and my current research is, as we're talking about today, bottlenose dolphin social interactions. As Teresa mentioned, I'm also the host and producer of Aquedox podcast. So if you like what you're hearing today, you want to know more about it, you're interested in aquatic veterinary medicine, I see some of you on the call have actually been on the podcast as guests. So thank you for being here today to support me. But it's a cool way if you're interested in learning more. I put up episodes about once a week. Also, I'll plug this again at the end, but if any of you are doing cool research that relates to aquatic animal health and you want to share it to a larger network, we reach hundreds of listeners across 95 countries every week. So would love to talk with you further about that. And I'll have my contact info at the end of the presentation. Great. So this is our first Poll Everywhere question, and I can see there's already 29 of you responding. So thank you for playing along. So I'd love to get to know who you are, who's here today. You can answer more than once here. So if you identify with more than one category, please feel free. We'll let it, I see the numbers are climbing, so we'll let this sit for a moment or two, and then we'll get to see where people are. I'm definitely B and E. I am a veterinary student. I'm also a huge marine mammal enthusiast. So let's see how this is going. Excellent. Wonderful. It looks like we've got a pretty good diverse group today, which is great. I love all the enthusiasts in the room. I hope you stay enthused throughout this presentation. Great. This is the next fun one. Oh, look how cool that is. So I'd love to know where are you right now? Where are we? We're in all these different time zones. And I think it's cool for us to just get a perspective of where we're all located. Oh, look at that. They're all popping up. This is so exciting. I don't even know what time it is right now in Africa, but thank you for being here, even if it's in the middle of the night or other places. I do appreciate that. Very cool. A lot of East Coasters. I'm also on the East Coast in the United States right now. So cool. I hope you all enjoyed that. I really enjoyed that. Great. So this is what we're going to talk about today. This is the paper that we're going to be discussing, and it should have open access right now in Marine Mammal Science. If you have any issues, just let me know. It was published last year, and like I said, this was the culmination of my undergraduate thesis research, and I graduated in 2018. So let's talk a bit more about dolphin associations. So bottlenose dolphins live in complex fission-fusion societies where social interactions and relationships between different individuals within that society contribute to their overall survival. Now, groups can merge and split over time, making group composition and individual associations a dynamic property. Yet, even within these large fluctuating networks, dolphins in Sarasota Bay, which are the dolphins we're going to be talking about today, form more stable social units that can vary based upon an individual's home range pattern, foraging need, life stage, and sex. Now, for example, some of these more stable units are adult males. They form long-term alliance pairs that move between adult female groups. Juveniles will often associate in fluid mixed sex groups, and then adult females try to maintain close associations with their recent calves, as you can see here in this image with the mom and calf. They also tend to associate in larger nursery groups. So those are comprised of females of similar reproductive status. Now, looking specifically at the alliance partners, while both paired and non-paired males successfully sire calves, it's actually been found that males in Sarasota Bay that do have confirmed alliance partners sire proportionally more calves than unpaired or solitary males. And on the flip side, with the females of similar reproductive age tend to associate with one another to increase their reproductive success. And they do that by forming a larger defense against predation, and they can learn from more experienced females. So other reasons why dolphins might want to associate in groups would be for reduced predation, increased foraging efficiency, enhanced defensive resources, and also the possibility of social learning. Now, because these repeated interactions are important indicators for survival, events that can separate an animal from its network of associations, such as an injury, one could say, might pose a risk to the well-being of the animal. So close interactions with humans put dolphins at risk of injuries, specifically from boat strikes and entanglements in fishing gear, which is what we're going to be talking about today. Now, there's previous studies focusing on the dolphins in Sarasota Bay that look at boat strikes and fishing gear injuries and determine that they can harm an individual due to lacerations, loss of mobility, interference with foraging, and can also affect dolphin behavior, specifically by causing changes in dive length, foraging habitat selection, whistling patterns, and surfacing patterns. So for this study, we applied social network analysis to gather a more complete picture of how an animal fits into a dynamic community. And given that injuries are known to impact the overall fitness and behavior of bottlenose dolphins, we hypothesized that animals suffering from injury will have fewer associates. Sorry, there's a motorcycle brigade outside. Hopefully you can still hear me. We'll keep towing. You can't interact, so I hope you can hear me. But anyway, we hypothesized that injuries are known to impact the overall fitness and behavior of bottlenose dolphins, and that animals suffering from injury will have fewer associates compared to non-injured animals and will exhibit lower network connectivity and greater solitary behavior. So that was our plan going into this project. So now let's talk about how we actually went about doing this. So the Sarasota Bay region of the central west coast of Florida encompasses Sarasota and Manatee counties and is home to a well-known long-term resident dolphin community. So this is just a little bit closer picture here of the Sarasota Bay area for those of you who might not be as familiar with it. Now, the Chicago Zoological Society's Sarasota Dolphin Research Program has studied this community since 1970 and actually just celebrated their 50th year last year in 2020. And what they do is they document characteristics and life history patterns of individual resident animals through photo identification and temporary capture release efforts. This is the world's longest running study of a wild dolphin population. And specifically, the program focuses on research and conservation projects that contribute to a better understanding of the social and dynamics of small cetacean populations, as well as the natural and human-induced factors that impact them, which is more what this study is specifically focusing on. Now, there have been hundreds of studies coming from this area in the Sarasota Bay Dolphins, and it's truly a really great resident population to learn from. So I just pulled a couple of the more recent papers here, but if you're not familiar with this population, though I'm sure most of you on this call are, there's so much out there to learn from them. It's really incredible. So for those of you who aren't as familiar about how we identify dolphins, what we do is we use photographic identification based on their unique dorsal fin and body markings. So essentially, just like a thumbprint, you and I have different thumbprints. It's how you can identify us. Their dorsal fins, which is what you can see here, are all unique as well. And we can make certain markings on them as well to help with that. But researchers who are there long-term can identify these animals really easily as specific individuals. So current estimates indicate that in the Sarasota Bay area, there are 170 known dolphins spanning as many as 5 generations, and they all live within about like 125 kilometers squared area. And 96% of these animals are easily identifiable, and over 90% are of known age and sex, as well as matrilineal lineage, maternal lineage, paternal lineage, or a combination of the two. So it's amazing. These animals are so well studied, which is how a project like this can happen, because we had to monitor these animals over an extended period of time and see not only what they were doing, but who they were interacting with, which required also knowing, those animals' identities as well. So for this study, we looked at 24 bottlenose dolphins. They ranged in age from 1 month to 42 years. And each animal was paired with a control animal that matched its sex and age class at the time of injury. So as you can see here on this slide, we have calves, we have juveniles, adults, and then adults with calves. So adult females who still have their calf with them. And why we paired them with control animals was essentially to establish a baseline for the behavior of an individual, and also to minimize the number of external factors that could potentially contribute to differences in associations. So looking specifically at the sex of our animals, we had 13 males, 10 females, and the sex of one of the dolphins was currently unknown. Now, as indicated, we looked at 2 different types of injuries. So animals that were entangled, often in fishing gear, and then those that were struck by a boat. Now, you can see the image on the left is an entanglement, and entanglements were identified based on observations of hooks, lures, monofilament, crab trap float lines, or potential scarring patterns that are consistent with wounds from lines. On the right side of the screen, you can see boat strikes. So those were identified based on the occurrence of deep, evenly spaced parallel cuts, kind of like what you can see in this image, or other indications of severe blunt trauma. Now, we didn't consider any individuals in our study with crescent-shaped scars or wounds. These are more consistent with shark jaws and could have been shark bites. So if we weren't certain, we didn't want to have that confounding factor. So more boat strike, the propellers, like what you can see in this image here. So for our study animals, we had 9 animals that suffered from a boat strike, 14 from entanglements, and 1 animal sadly had both a boat strike and an entanglement. We also looked at intervention, which was an important component of this study. So 11 of our study subjects had an intervention, and was particularly those that were free swimming dolphins with potentially treatable active entanglements. And the way that we define intervention is essentially any rescue attempt by humans in which an animal received assistance in the water or in an on-land facility. So this image here that you can see depicts a temporary capture and release rescue observation that was done by the Chicago Zoological Society's Sarasota Dolphin Research Program with approval from the National Marine Fisheries Service. So just one example of a type of intervention. Now, we applied the half-weight index, which is the current industry standard for associations between dolphins. Essentially, it's a fancy calculation that tells you how can you define an association, and it looks at how many times and how 2 individuals are interacting. The paper has the full calculation if you're interested in it. We can also talk about it afterwards as well if you want more information, but this is typically used for bottlenose dolphins. Now, we had a couple different networks, so I really wanted to take the time to parse them out because we had 4 different networks that we were looking at. So this first network is the full association network. So any interaction between 2 individuals in the entire population with that half weight, which is how we're defining our association, equal to or greater than 0. So equal to 0 would mean they actually, two individuals never associated during a time period. So, but that animal was included, meaning that like if you are dolphin A and dolphin B happened to be in the population at the same time, but dolphin A never interacted with dolphin B would still be considered as part of the full network. Okay, that's our full network. Then we looked at preferred associations within that full network. So the way that we do that is we find the mean half weight of that full network, which was 0.03. So 3% of the time they're together. And you double the mean, so 0.06. And any interaction with a half weight greater than 0.06 gets to be considered in that preferred association. So who are the quote best friends within that population would be dolphin A and dolphin B, who has a half weight value greater than 0.06. I'd ask for head nodding here, but I can't see you. So hopefully you're all following with me here. So then we have our filtered network, and the filtered network differs in that it only looks at half weights that are greater than 0, meaning that if dolphin A and dolphin B did not associate at all during the time period, dolphin B does not get to remain a part of the network. And what's cool about filtering here is that essentially you're just looking at the true interactions between dolphin A, your study subject, and everyone else in the population that dolphin A actually interacted with and was cited at some point during that time period. Now, we then filtered the filtered network further down to preferred association. So we did this exact same thing as our original full network. And we said, what is the mean half weight in this filtered network, which was 0.07. And then to get that preferred, we multiplied that mean by 2 to get 0.14. So this is what we're going to really be looking at as we approach our results. So essentially, who are the quote best friends in this population? In the filtered network, it's those dolphins that have a half weight value between them. So dolphin A and dolphin B with a half weight value greater than 0.14. So they were seen together in all sightings during that time period, 14 or more % of the time. I hope that makes sense. If it doesn't, in the question and answer, we can go through it again. We're also going to be looking at some, this is a network graph. So I just wanted to familiarize you all with it now. So the dots here, which are nodes, refer to individual dolphins. So dolphin A, dolphin B, dolphin C, dolphin D. The lines, what are called edges, demonstrate an association. So if there's a line between two dots, that means those two dolphins associated during the time period. And the thicker lines indicate that it was a higher half weight, so they associated more frequently. So, the one towards the top could be I'm making up numbers here, but could be like 0.67 versus a really skinny line could be 0.02 or something like that, if you get the idea. So we also looked at 4 different social network measures. So we looked at strength, eigenvector centrality, clustering coefficient, and the number of triangles. Now, strength is the interaction rate. So essentially, how many individuals is a dolphin associating with in a sample period? Eigenvector centrality is the sum of the centralities of an individual's neighbors. So what's useful about this is that it actually captures the importance of individuals in the network as like social hubs, you could say. So it's useful for looking at propagation of information or diseases through animal populations. Thirdly, we looked at clustering coefficient, which is a measure of the degree to which nodes in a graph, so the dolphins, tend to cluster together. So how well are associates of one individual associated together collectively? And then lastly, we looked at number of triangles, which refers to the connections between nodes that complete triangles. So essentially, if dolphin A is associating with dolphin B and dolphin A is associating with dolphin C, are dolphin B and dolphin C also associating to form that triangle? Hopefully you got that visual of a triangle in your head. Great, so those were our 4 network measures that we looked at moving forward. So now let's look at our results and see what actually happened here. So the first thing we wanted to do before we got into all the network measures was just look at the overall group size. So did the overall number of dolphins that an animal was sighted with change after it got injured? And what you can see here is that we found actually no, they didn't change. The group size remained pretty consistent before and after injury, both for our control animals, meaning they weren't injured at all, but we just used the time periods. And then for injured animals as well, we didn't see any differences. Great, so it means that the overall number of dolphins is pretty consistent after injury. So then we looked at that unfiltered network connection. So that first full network we were talking about, where the half weight was equal to 0, so including those that our focal dolphin that we were studying might not have associated with. And then the right side of the table here is that preferred unfiltered network. So again, looking at who are the quote best friends in that full network. And we found here is that there weren't really any differences between before or after injury for our animals. However, when we looked within those filtered networks, so that second group of networks that I was telling you about, where we took the half weight had to be greater than 0, and then we filtered that further to our preferred network. So this is looking at the quote best friend, so that half weight greater than 0.14. We actually found that those metrics that I was telling you about before, so strength, eigenvector centrality, clustering coefficient, and number of triangles, all decreased after 1 year post-injury. So visually, what that means, because I'm a visual person, I need to see things, numbers always sometimes don't make sense to me. So on the left here, you have a representative animal, FB09, who had a boat injury. And this is her preferred network before her injury. And then on the right, you can see that there are fewer nodes, so fewer dots there, meaning that there were fewer preferred associates, so those fewer best friends, you could say, after her injury. So hopefully this visual helps you visualize like what that result meant. So then we wanted to see, well, what happens further down the road? We wanted to see, could a longer time period potentially be sufficient for an injured animal to rebuild its social network? And what you can see here, again, in the blue is our p-values. And we actually did find that there was a trend towards returning to preliminary network metrics. They didn't return to full baseline, but what we were able to find was that you could see some of them. So we noticed that there were these differences, but we wanted to know, do these differences relate to the sex, age class, type of injury, or presence of intervention? Because that could have been confounding factors in our observed network changes. So what you can see here, again, the p-values are all boxed in blue, and we didn't actually see any differences with respect to any of these, which is great. It's more indicating, as we'll discuss a little bit later, but that it's more the injury itself rather than these other factors that might be causing these behavioral changes in the dolphins. So with these decreases in network metrics for the preferred association networks, we were interested to see if the most stable pairing structures that we might have referred to earlier in this presentation would remain despite this injury. So I have another fun question for all of you. Hopefully it's popping up. And so we're going to talk about, well, what we noticed that there were these network differences. There was a decrease 1 year after injury. So what associations do you think remained? I will tell you that there might have been some. There is more to this talk, so that might help some of your answers here. So was it just mom and calves that maintain their associations? We talked about at the beginning of the presentation that they're very strong associations. Were they just male alliance partners? Again, a very strong association. Did both of them keep their alliances and their associations despite injury, or did sadly none of them get to keep them, and injuries just are really bad? Don't get injuries. This is why we have veterinarians. My job security here that will fix the injuries and hopefully they can get better. Looks like we've got 24 of you. Oh, I love poll everywhere. It's fun when you're on this side. As a student, sometimes it's not as much fun and it can get stressful. But I hope again, I can't see you, but I hope you're all having fun with this. Maybe we'll see if we can get to like 26 before I go on.
Michelle, I'm so sorry to interrupt. We're not seeing live updates on the screen. So I don't know if we're not seeing the results that you're seeing.
See them now?
For me, it's blank. I don't see any responses.
Oh, you didn't even see the graph ones too? Oh, guys, that's so sad. I'm sorry. It's coming up over here, and when I practiced it, it came up. Well, what your group has said, 44% of you have said mom and calf, and 56% of you said all of the above. Oh, somebody's changing their answer. So about 40% of you think just mom and calf remain strong, and about the other 60% of you think both mom and calf and male alliance partners remain strong. Bummer you can't see that. That's unfortunate. Oh well, here we go. So for those of you that chose mom and calf, you are absolutely correct. They did remain strong. So in 6 of the 8 documented cases in which calves were injured, the mother remained the strongest associate before and after injury. So 6 of the 8, that was great. For the other 2, one of the calves wasn't cited after injury, and then the other wasn't cited after injury with anyone above 0.14. Similarly, on the other side, looking at mothers who were with calves who got injured, the calves remained the strongest associate both before and after injury. And in the third case, so 2 of them had remained the strongest before and after. And for the third mom with calf, her calf remained one of the top 5. So very clear continuity there. And what you can see in this figure, I know it's a lot, so bear with me, but essentially I want you to just focus on the dot that looks like a target, so a little black dot inside the white dot, and then look at the open white dot. So the open white dot is mom, and then the target dot is the calf. And so this calf was only 1 year old at time of injury, and it was just kind of like an ideal representative calf. So our top row here is our full network not filtered. So that's why there's a lot of lines and a lot of animals there. Our bottom line, our bottom 3, so D, E, and F, are all our filtered networks. And so the ones all the way on the left here are the 1 year prior to injury. And then the middle is going to be 1 year after injury. And then all the way on the right is 2 years after injury. So if we're focusing on this bottom row here, D, E, and F, what you can see is that this is our filtered preferred. So our 0.1 greater than 0.14 halfway. So our best friends here. And really what it comes down to is that you can see a line between both of the white circle and then the target circle in all 3 of these graphs. And it's actually a pretty solid line. I know it might not necessarily come across on your screen, but it's a pretty solid line, demonstrating, again, when we talked about before, that the thicker the line, the stronger the association. So those were definitely maintained after injury and 2 years after injury. So in Sarasota Bay, calves are closely associated with their mothers for the first few years after birth. They require nutritional support from their moms for at least the first year, though many tend to associate for about 3 to 6 years before they're weaned and they associate more with the juvenile grouping. Previous studies suggest that the majority of the behavioral and social learning in bottlenose dolphins occurs from moms teaching their calves. So that might be one reason why these relationships are so stable and maintained. Now, for those of you who chose answer, I think it was C, both, that was actually the correct answer to our question. So both moms and calves and alliance partners actually maintain those relationships. So congrats to the 60% of you. The other 40% of you, I hope you learned something here. This is great. This is new knowledge, very exciting. So in 2 of the 3 documented cases that we had with males confirmed alliance partners, the strongest relationship was that alliance partner. And what's really cool is in one of those, the half weight, so that association value before and after injury was equal to 1, meaning that 100% of the time that this dolphin was sighted, it was seen with its alliance partner. And all of the other relationships that it had in associations were less than 0.25. So it's very evident in this case that, or the strong suggestion that A, they're alliance partners, and B, that they're together like all the time. So it was super cool. And then that third male that had an alliance partner, the alliance was the strongest association before injury, but the injured animal didn't really survive much after injury, which was a potential reason why they didn't continue associating, quite sadly. So a similar graph to what we just looked at for moms and calves, here is alliance partner. So again, the top is our unfiltered, the bottom is our filtered networks, and then the left column is before injury, the middle is 1 year after, and then the far right column is 2 years after injury. And what you can see here, similar to mom calf, is that the line between the open dot and the target dot is quite strong, especially if you see 1 year and 2 year after injury. Those are more blown up figures. That line's really thick. So clearly indicates that these animals were associating and quite strongly before, after, and 2 years after injury. So the relationship's really with the longest duration in Sarasota Bay, especially because mom calves, calves typically wean after a couple years. And so they do associate, stay tuned, some other research I'm working on. They do continue to associate after they wean, but really these longest duration ones are the male pair alliance. Most of these alliances are formed around sexual maturity, though there might be some new research coming out shortly that would indicate earlier, these alliances might form slightly earlier than you would potentially think about. And in Sarasota Bay specifically, more than 93% of males have typically formed alliances before 20 years of age. And dolphins can live much longer than that, as many papers out there have documented. And so these bonds typically are maintained through life or until one of the pair dies. And that was a really important part for this study, particularly because if injury was interpreted as a sign of either impending death or death with the potential to remove an animal from its population, what would its alliance partner do? If it incorrectly assumed that the partner had died, then they could have sought off a new bond, and it could have completely disrupted the social association between these two animals. We weren't really able to see much of that in this study, just because the 2 confirmed males with alliance partners didn't receive any type of intervention. So they weren't necessarily physically removed from the population. So it would be interesting to see what would happen if one of them was physically removed in this study. Another study, though, did look at 2 bottlenose dolphin males in the Indian River Lagoon, and so another part of Florida. And they were separated for 6 months due to a rehabilitation. One of them had been moved to an out of habitat rehab facility and came back, and they reassociated with one another. So that was really cool to see that they could still bond and still maintain that association afterwards. It's unclear though, like how long an intervention would be. If that animal had been out for 2 years, would that change things? Who knows? If there's newer studies on that, I'd love to hear about that. If not, it's something that as situations arise, we'll just have to continue to monitor and see case studies of like what specific individuals do. But I think male pair alliances are super interesting, super cool. If you haven't heard much about them, you should definitely go read up on them. They're fascinating. So that was pretty much the bulk of what we looked at. So let's look at our main takeaways here. So what we essentially found was that while injured animals were found in groups of approximately the same size prior to injury, the composition of those relationships, particularly like those strongest relationships, that halfway point over 0.14, varied. And so within that, we had strength decreased for individuals, suggesting that the animal had fewer preferred associates despite maintaining that larger, like wide population community. And then the decrease in eigenvector centrality and clustering coefficient that we discussed suggests that the injured individuals, they can associate with a greater variety of less connected individuals. So they decrease their global reach in the full population. And then when looking at the decrease in number of triangles, what that implies is that injuries might lead animals to be found in more isolated and less tightly knit groups. So part of our hypothesis was that injured animals would exhibit greater solitary behavior. And what we found was that on the whole full population, not filtered, that wasn't the case. But when we looked at those best friends, that preferred network, we did see that they did have more solitary behavior. So some interesting differences than what we had initially assumed. But the good news is that we found that after 2 years, the animals continue, though they do continue associating with a greater variety of less well-connected individuals, and they don't necessarily return to their full pre-injury network values, but we did see a positive trend suggesting that there is opportunity for post-injury recovery. And what would be interesting to do would be to look 5 or 10 years down the line, should the sighting network data allow that, and see what happens specifically with these animals again on a longer term, and see if those pre-injury values are fully back, or honestly, another totally different project would be, do those main associations change over time as an animal changes in life stage. So that would be interesting as well. And then we talked about, we looked at the age, sex, type of injury, and presence intervention, and we didn't see any relationship between that and our changes in behavior. So to me, what that implies is that it's the presence or potentially the severity of an injury that's going to have more of an implication on the behavior rather than these other changes. And I did want to draw attention to intervention because what our results demonstrated was that interventions don't negatively impact the social connectivity for an injured dolphin. And considering just how important interventions are for the survival of bottlenose dolphins, they're so important to maintaining the populations and doing great things. So our results provide additional encouragement for rescue teams to continue their amazing work and providing assistance to injured dolphins, especially since the injuries from this paper were looking at anthropogenic causes. So those are human induced, so boat strikes and entanglements. So really, really good takeaway there. Now, there's a couple reasons why these behavioral patterns might exist. One potential reason from the injured dolphin's perspective is that it might be its attempts to maintain the size of its larger network by having more variable and transient associations. So if its best friends are decreasing, maybe just having more friends overall who change over time might be a useful thing for their fitness and overall survival. On the flip side, you can think of the non-injured dolphin perspective, where they might interpret injury as a sign of weakness, and so therefore might be less likely to associate with an injured animal repeatedly. So that's going to then increase the variety of associates for the injured animal. So two interesting thoughts there. What's super cool is that though the second interpretation from the non-injured animal perspective, well, it hasn't necessarily been studied yet in marine mammals, which would be cool. If you're doing that, let me know. But other species, such as ring-tailed lemurs, actually can distinguish weaker individuals through changes in their pheromones, and then they can adapt their behaviors accordingly. So it has been documented in mammalian species. So it'd be interesting to see if there's ways that we can identify that and study that specifically within bottlenose dolphins. And our findings really conclude that injuries do disrupt social relationships of animals. And so because these dolphins rely on these associations for survival, increased occurrence of injuries from boating or fishing or things like that can actually put these animals at greater risk for long-term survival and therefore make them more vulnerable to predation. So some suggestions to how to reduce the frequency of these animals incurring anthropogenic human-induced injuries could be to follow this marine life viewing guidelines and dolphin-friendly fishing and viewing tips, which are here, and there's also a link in the paper. And so, some of these might seem common sense to some of you, but for a lot of people who aren't necessarily in the marine mammal community, being able to remind people of this and being mindful of it are actually really important. So things like, throwing away your trash, staying at least 50 yards from marine mammals, avoiding excessive speed when you're out on a boat, or try to change the direction, go away from the dolphins if they're near you. If you're fishing, take your fishing gear with you. Don't leave extra line. Little things can actually make a huge difference and can mitigate potential for dangerous human-dolphin interactions, and can hopefully go a long way to save these animals. So just keep these in mind. There's lots more tips out there. Just I think education for everyone is always a really important thing. So that's just a plug for, if you see people out there, just be very nice about it and just try to educate people, because they might just not know, and you can help the dolphins and other animals in the water as well while you're out there. So with that kind of brings us to the end. I'd like to thank the Chicago Zoological Society and the Sarasota Dolphin Research Program, as well as the Princeton Environmental Institute and the Princeton Department of Ecology and Evolutionary Biology for supporting me in this project over the past few years. They've all been wonderful and have really been instrumental in helping me grow and learn as a researcher. So I'm truly thankful to everyone. And with that, I will take any questions. My contact info is here. I've got my email there, as well as social media if you want to reach out to me. As I mentioned earlier, I'm always looking for new people to interview for the podcast. So if you're research, or if you have a colleague who does research relating to aquatic animal health, you do not have to be a veterinarian. You don't have to be a PhD. You just have to do something or know someone that does something within aquatic animal health that you think the public and your colleagues should know about. Please feel free to send me a DM or email me, or if you have any questions about this or other things. If you're a student listening and you're like, how do I go and be an aquatic vet or do stuff like this? I'm always willing to talk with students as well. So please feel free to reach out. And Teresa and Eric, if there's any questions, I would be happy to take them.
Great. Thank you so much, Michelle. That was fascinating. So yes, as a reminder for folks, if you have questions, please use the Q&A function that's at the bottom of your Zoom screen to post those questions. Eric is going to ask those in just a minute. We're going to let a couple more come in. Looks like there's already some, but I'll start off with one. So you showed us the sample sizes of the number of dolphins of different ages and different sexes that you used in this study. Is that all the dolphins in the Sarasota community that have experienced some kind of entanglement or boat strike, or was that a subset of the animals? I guess what I'm asking is, like, how prevalent are these kinds of injuries in the wider population?
Sure, good question. So as far as I remember correctly, this was a subset of individuals. We really wanted to make sure that we could correctly identify what the type of injury was and be able to classify it into either an entanglement or a boat strike. And we wanted to make sure that we had a good sighting history for these animals, because the study was so important, looking at before an injury, after injury, and then 2 years out. If they weren't cited, more than a few times, I think in the paper, and I'd have to go back to confirm, but I think we said 10 times. It was either 5 or 10 times, so they had to be cited in each period to be included. So if there was an animal that was only cited once, we weren't really going to be able to get a representative idea of who it associates with over an extended period of time. As for the exact percentage, that would be a great question for Dr. Wells. I'm just not as familiar with the full population data set.
That makes sense, but that's interesting to know. It's potentially a subset. And then related to that, and this might not be something you can answer because of what we just talked about, can you say anything about whether males or females or young or older calves were more likely to have those kinds of injuries?
It's a great question. I don't think I am personally qualified to answer that one, but I would refer you to Dr. McHugh and Dr. Wells, and there might be papers out there on that. I just not certain.
No worries. Eric, do you want to go ahead and ask some of the questions that we're getting in the Q&A?
Absolutely. Sounds good. And thank you for the wonderful talk, Michelle. All right, I'll start with, you have 6 questions so far. So the first question is from Jennifer Volvo. In the male alliance pairs, does one of the pair sire offspring more often than the other? With this population, how related are the males in the male pair?
Thank you, Jennifer. That's a great question. These are all fantastic questions that I wish I knew more about. I don't know if it's one male or another. I personally haven't done a lot of studies specifically on the male alliances. I think they're fascinating, and I've looked more at just continuity in relationship. I know there's a lot of papers out there, so I'm happy to like look into that and get back to you, or maybe there's somebody on the call who studies that, but I'm not entirely sure. And then there was another half to your question, it just went away. Oh, how related are they? Again, a great question. I don't know. I will say that I've been working on a different project, looking on more the Eastern dolphin, the IRL dolphins in Florida, and we're looking at kinship with respect to associations. So that should hopefully be out in the coming months. So stay tuned on that, and that can hopefully answer a little bit more about that. But I don't know if we specifically looked at male pair alliances, but it did look more at kinship with respect to associations. So sorry, that's not really that helpful.
Okay, we'll get to our next question. Jorge Catanave, is partial or full amputation considered an injury, as it is something permanent and won't heal? So yeah, is it partial or full amputation as an injury? I guess, yeah, if it's permanent, does.
It's a good question. So I think, so with respect to this study, right, we were looking at injuries that we could identify from either a boat strike or an entanglement. So if we knew that that injury as an amputation, like was something from one of those two, it could have been included. I don't know these animals specifically well enough to know if any of them had that. I don't believe so, but don't quote me on that. But like, yes, an amputation would be considered an injury. I would refer you if you are interested in that type of stuff. One of the dolphins that was rescued, sadly, she passed away in November. But Winter was a dolphin at Clearwater Marine Aquarium in Clearwater, Florida. And she did lose her tail. So would have been considered an injury. And it was permanent, didn't heal back, and was able to still survive. So I think there's actually going to be a lot of interesting papers coming out in the coming years, looking at her and her morphology and like how that affected, though she was brought into managed care facilities. So like a study like this, looking at wild population associations, wouldn't necessarily be practical for her. But if you're interested in more the like anatomy, physiology about losing limbs, or she didn't lose a limb, she lost a tail, but you know, similar thing, would recommend that you look into her. There's a lot of interesting articles about her of late in the news.
Thank you. Okay, next question. Mariana Chavez Andrade. Very interesting talk. Congratulations for your work. Which do you think, or what is your main hypothesis about the decreasing interactions after injuries occurred?
I see, before I answer that, I'm just going to refer to the last question. There was a comment about seeing amputations in orca and boat strikes for orcas. So super fascinating. I want to hear more about that. So we should chat after. And then this question was referring to main hypotheses. So I think the main hypotheses are kind of the two that I brought up. So one, like, is it an injured animal who just like isn't able to interact with those same animals. And so is just like, I'm going to interact with anyone who'll talk to me right now because I can't keep up. Maybe it's a physiological thing. Like if they have an injury and they just can't keep up with their peers, then, maybe it's that thing. It could be the other perspective was, from the non-injured animal side of things, where maybe they're viewing it as a sign of weakness. And so it's not the injured animal's fault. It's actually the rest of the community who's viewing that animal as a sign of weakness. And they don't want to, even if they're not like actively thinking about it in this way, maybe they don't want to decrease their fitness. So I think those are some of the thoughts, though there could be others out there, and I'd be interested to see what other people think. I think those were the two that we were looking at, though, potentially there's more out there with respect to, different ways that you can construe it. I don't know how we're going to get an answer, right? Like, I think we have to be able to communicate with them in order to do that, which would be fascinating as a whole other talk. And I'd love to see that. I just don't think we're there yet. But yeah, I think those were like the two things that we were thinking about.
Great. Next question from Isabel Rosado. Do you know what are good sources for learning more about this topic?
Yes, what a great question. So if you're interested in social network theory, like at large, specifically within animals, there is a book that Dr. Hal Whitefield wrote. It's called, I think, The Animal Social Network, or Animal Networks. I cited it in the paper. Also, if you send me an email after, I can make sure that I send you the Google Books link to it. It's an amazing book that really walks you through how social networks and animals and how things work and how things change. If you're looking more from the dolphin side of things, specifically within this population, there are some other studies that look at interactions that I'd be happy to refer you to. Off the top of my head, I can't think of them, but there's definitely others that I cited a lot of in the paper that you can refer to. And again, send me an email and I'd be happy to send you some of those papers.
Great. Next question is from Jessica Taylor. Do you have some insights into how losing an alliance member affects the rest of the social network?
That's a great question. Wow. I wish. I think it'd be really interesting to look at because, right, like if an alliance partner, taking the ones that I mentioned before, where their alliance was 1.0, meaning they're together 100% of the time, and the rest of the interactions were all less than 0.25, I think it's really going to affect like all of those metrics and how it's interacting. Because if you're losing, in human terms, colloquially here, right? Like if you're losing your best friend and now there's all these like other, associates that you've had, maybe you go spend more time with them now because you lost your best buddy. Maybe instead, as we were kind of hinting at with the loss of an alliance pair, like maybe a male will go find a new one, in which case like it actually wouldn't affect the rest of the network, and it would just, one dolphin leaves and a new one comes in. Maybe it's not 1.0 the first year, maybe it takes a little bit to build up. Yeah, cool question. Interesting to think about. I will have to think about it further, but those are kind of off the top of my head, the thoughts that I have on that.
And I'm sure it varies a lot by population, depending on, how many animals are dependent. Like from our work in Belize, the population is much smaller, but, these animals have been together for decades. And, if you lose an animal, I think kind of like elephants, it's not that strictly in this structure, but, if you lose the animals that had the knowledge and the adaptability to deal with the last 40 to 50 years, I can't imagine that you're not losing some kind of like important information from your social network, or like at least losing skills that were important for you too,
Yeah, I mean, I think that's why this type of analysis is so interesting. Just because like there's so much you can gain from it, right? Like what if you remove one pond, then what happens? You can recalculate everything without that one individual. Or what if you look at an even smaller subset of individuals? Like I said, right now I'm kind of, this next project that I'm working on is looking at kinship, and more specifically on like mother-calf relationships and stuff. And like that opens a whole new area of who's associating with whom, how does kinship and familiar relation get in there? We as humans think like, oh, family, is for life, but like, is it with animals? In some, like elephants, you're saying, right? Like that's very, very much like that. And, some orca groups as well. For dolphins, is it not? Like, oh, there's so many things.
Lots of things to solve. Okay, next question here, and I'm going to combine these two because I think was his intention from Jorge Catanave. Are there any comparable studies with sick versus injured individuals and their interactions? And then he said, we see amputations in orca from boat strikes. That's why he asks.
Okay. I think, and you can type back in here. So are we saying like sick as in more like an internal disease that you can't see versus injury as something that's like visible from the outside, like an amputation or a laceration or something like that? Is a comment back? Disease versus external. Okay, cool. Yeah, so, right, it's really hard. Also, sorry for the dog barking. My roommate just got home with the doggy. It's a cute doggy. Anyway, it's an interesting question, right? So when you're talking about sickness in a wild population, how do you know if an animal is sick, right? Like if you can't visibly see it, then how can you know? If you visibly watch a boat impale a dolphin and there's no external injuries, odds are there's probably something inside of it that's not working out well. And you'd assume that there's injury, but you won't actually know until you get into it. And most of the way that we see internal injury is actually on necropsy once animals strand. So it's an interesting question, and it's something that I thought a lot about with this paper is, are we missing a lot of animals here that have internal disease? Maybe, yeah, like there's a very real possibility. But then also when you're talking about internal disease, is it necessarily going to be from like a boat strike and entanglement? Maybe not. It could have swallowed something that we wouldn't be able to see. It could also be, through pollutants in the water and stuff that would be potentially, anthropogenic, human induced. But there's also like other physiological issues that could occur, which would be totally natural, which would then also potentially disrupt their associations. So it's a good question. I think that right now we don't have the means fully to determine that, but it's something that we should work towards, and through capture and release health assessments, we can get a taste of what's going on. But you can do blood work, you can do lots of things and figure out what's happening in a body system, but you still might never know for certain what's actually happening. So great question. I'd love to know. I think we've got a long way to go before we can get full answers to it.
Agreed. Next question from Jennifer Volvo. Is network analysis continuing to possibly beef up sample size, as well as possibly enable the investigating the impact of the severity of injury?
I assume you mean with referring to this specific study. The short answer is at this second, no, but I think that it would be something worthy of looking into in the future. I would like to see, down the road, how do things change as these animals continue aging? I'd have to look at the study samples again and make sure that, which animals are still alive and can we continue monitoring them? I think it would be really cool to circle back to this in like 10 years and see how does that change, or, pivot from this. And you mentioned severity of injury, looking just specifically at that. That's hard because like retrospectively, we'd look at photos and looking at severity, we obviously have to come up with an interesting scale. So yes, I think it's interesting. I think that maybe once I get my degree and don't have exams all the time, like all day yesterday, I'd have more time to focus just on that. Or if anyone here wants to collaborate with me on it, I'd be totally down. I think it's worthy research and would love to do it.
Good answer. Okay, next question from Reina Doracio. Were you able to account for the distance that these animals traveled before their injuries? I.e., was a dolphin who tended to remain within a small area before its injury more likely to maintain its association better than a dolphin who tended to travel over larger distances pre-injury? You might have to read that one yourself once or twice.
Yeah, I'm gonna read this. I also just realized it got dark because it gets dark now at like 4:30. So I hope some of you could see me during that presentation. Just put on a light. Were you able to account for the distance that these animals traveled before their injuries? Oh, okay. Okay, I think I get what you're asking. In this study, we didn't look at spatial area. There are really cool studies out there that look at the specific areas, and they use a lot of cool technology that I don't quite understand yet, how to track them over areas. So yes, it would be a useful thing to look at, seeing did the specific areas change. We did essentially account for, a study area. We looked at from, I think, like Tampa Bay to Venice Inlet. I think those are the proper words, though. You can look at the first figure in the paper to look at the exact area. And so we know that dolphins were in that. We didn't specifically track like where that one dolphin went over time. If that was your question, I hope I answered it for you.
Okay. Next question from Kirsten Donald. Would it be possible that the lack of association with animals on the fringes of its network in the community is the injured animal's choice? Not feeling like associating with a lot of individuals outside of your closer knit ones because it was not feeling good or confident. Like when you're sick and you only want to see friends and family, but you don't want to hang out with others until you feel completely better.
Great question. And I really appreciate the analogy. I think when you're doing network analysis, sometimes the terms get really big and you're like, okay, what happens in humans and how can we understand this? And then, things are different between different species, but like it helps. So I appreciate that, especially given everything. I hope everyone listening is healthy and doing okay and taking care of their family. Please be safe and healthy during this holiday season. Anyway, the question itself was, yeah, so it could be. What we found though was that like they were associating with more like fringing, and then right after injury, that preferred network, so the close network decreased a bit. So it'd be almost, at least what we saw, like almost the opposite potentially. Though I do think that I agree with you to some respect that like some of the very close associations that did stay, like might be that animal. And it's like, okay, I know you guys, like I only want to be with you. I don't obviously know what they're thinking. I do think that it would be a complete possibility that if they're consciously deciding who they want to associate with and the reasons behind it, I don't see why that wouldn't be a valid argument for it. So yeah, I think the logic behind it would be something I would agree with too.
Actually, I remember I was listening to a podcast the other day about something similar, and they were talking about how they do find that in people, some people are drawn towards like gravitating towards connections when they're sick, and others like completely spurn them. So sometimes people are like completely just need to be alone when they're sick, and that there's often a divide in like how it ends up. So I'm sure in dolphins, you get that same level of individuality.
Maybe podcasts are great. You learn great stuff on them.
Absolutely, right? Okay. Next question, Sabrina Croft. Hello, I'm a marine biology undergrad at University of Alaska Southeast. I was wondering if these types of stable associations, particularly male alliances, occur in other dolphin species besides bottlenoses.
Hi, Sabrina. Thank you so much for joining. Congrats. You're an undergrad. That's amazing. Do research, do cool stuff. Great question. To be perfectly honest, I haven't done a lot of studies outside the bottlenose dolphin population. Eric might know some stuff. I'm sorry to throw you on the spot. I do think they exist, but I'm not entirely certain. So I would err on the, I can find some papers for you and refer you to them, but maybe Eric has an idea.
I'm not terribly certain either, but I mean, the thing is that we don't know a lot about male associations in a lot of other species because we can't track them as long as we could bottlenoses, but in a lot of other, animals, the groupings, appear to be divided up, they can be matrilineal or patrilineal, but like, I'm not aware of a lot of data, I'm just not aware of it showing like stable pair male associations in a lot of cetaceans, although I'm sure to some degree, like in some work with steno bretonensis and rough-toothed dolphins that like I've seen, we've done like you see stable associations between animals, but knowing whether they're long-term with males is so difficult. I think it's probably true for other species, but not easy to identify.
Yeah, Sabrina, I think what's super cool about this population in Sarasota is that like Dr. Wells and his team have been studying it for over 50 years now. So, and there's a similar group in Shark Bay, Australia, that has been doing long-term monitoring as well. And so you can run a study like this and see the patterns of association amongst many other things that are studied in this area compared to potentially other species that we just don't have this for. And so like they might very well, but if we're not able to see them all the time, and like I said at the beginning of this talk, like 93% of this population is known and identified. Like that's crazy, right? Like think about how many aquatic cetacean species there are out there. Like some of the big ones like right whales, we kind of know who they are because there aren't that many, but this is a very special population because it's a resident population. So that doesn't necessarily answer your question, but I think we'll have to look into that one for you. Or if there's somebody else on this call that does stuff like that, you're welcome to chime in as well.
Okay. Another question from Jennifer Volvo. She says, sorry for another question. Very interesting topic. Did any of the injuries occur to an important individual, as in one that ranked high in the network score?
Interesting. I'm not entirely sure what you mean. Thank you also. I appreciate it. It was an interesting topic. I don't think we ranked like importance of individuals between, like if you're asking about like if one population, like one of their best friends or whatever, like also was injured, we didn't necessarily look at that. So like there are ways to take the full population and say who's like the most quote like social out of all of them that has the most associations, whether or not and then like looking at, are you like the social butterfly that everyone's friends with? We didn't necessarily look at it from that angle, though that would be interesting to see. Oh yes, the most social. Okay, great. Yeah. So we didn't necessarily look at it, but I think it could be interesting to see that.
Great. And the last comment and question here from Jorge Katsanave. Would brothers be considered a stable male association? And he's referencing two famous orca brothers who stayed together for over a decade.
That's cool. I'm gonna have to read about that. I don't know about those two famous orca brothers. So I'm not aware of any bottlenose dolphin literature that specifically looks at brothers as alliance partners. So I don't know for certain. I think the definition is that it's like two males associating on an extended period of time very highly. So like by that definition, potentially, personally, I don't see why you couldn't, though I'd want to confirm with those that study this like full time, how they would define that. I could say that in the other research that I've been doing looking at kinship, we didn't observe that for alliance partners were not necessarily like brothers. So, but that was also like, it's a small population, right? Like even when you're doing these great studies and we have this resident population, you're still only looking at, the study had 24 animals. So I haven't seen that, but that doesn't mean it doesn't exist. And it's something that I would want to look a little bit more into, but I'm very intrigued. And like I said, I need the two famous orca brothers. I need to read about that. So thank you for telling me about that. That's cool new research.
And he mentions here again that he mentions that it's in Valdez Peninsula, Patagonia, and that Dr. Holzel did DNA studies back in the 80s.
Excellent. Thank you. I love this. I'm teaching you, you're teaching me. This is brilliant.
And just to comment on that, I think, and correct me if I'm wrong, Michelle and Teresa, like with bottlenose dolphins, like in most places, it's not familial associations that are occurring so much as, you know, like in killer whale populations or other, you know, ones that are have more stable family groupings. Like by their nature, they're having more stable associations with their family, but that's the driving factor rather than, you know, where dolphins, it's not inherently the family group that becomes the associates. Is that that's the case still for most dolphins, isn't it?
Yeah. Yeah. So, I mean, dolphins are more like really define that fission fusion. So it's a lot of like dolphins all together, and it's not necessarily familial based like you would see with, you know, the orcas, for example. Though there are some relations that stay, but it's not typically, it's nothing compared to that, or like the elephants we were talking about before. It's not a similar thing.
Yeah,
Okay,
it depends
great.
with males and females, right? Because you see moms, long-term moms and their calves may associate more in large groups along with other unrelated individuals. But my understanding, this is going to be embarrassing if someone who's, you know, supposed to be a PhD in the subject, my understanding is that the male-male pairs tend to be unrelated individuals, like you were saying. They tend to be similar age groups, which often then is going to mean unrelated individuals because the same mom is not going to have two calves in like close proximity necessarily.
Yeah.
Okay. Michelle, that was fantastic. We are well over our time. We still have people listening in. So unless folks have any last questions, I'm going to throw out one more for sort of the students who might be watching. Since we are the student members, we have to ask these questions. And that is sort of a broad one that I'll let you answer as you like. We always like to ask our presenters about the path that took you to their current work, to help show students and other early researchers what's out there. And you have this really interesting perspective as someone who's in vet school, but also doing research, but also, you know, leading a podcast. So can you talk a bit about your path, sort of what drove you to the veterinary side of things? And then do you have any advice for folks who want to do the kind of work that you're doing?
Yeah, absolutely. And if there are any other, you know, fellow students on the call, props to you for being here. I know it's like exam period, and I know that there's a lot happening and you don't get credit for attending a talk like this, but this is how you learn about what's out there in the field and get to see what piques your interest. So my very basic short background is I was always really interested in marine mammals, and I didn't know where I was going to go with that. I actually thought I was going to get a PhD in marine mammal behavior. That was kind of my plan. And then I had an opportunity to do some veterinary work where I worked with a veterinarian, actually with penguins. And I just thought it was fascinating that she did so much. She could help return. It was a rehab program down in South Africa. And these injured animals would come in with like giant holes in them. For those of you who don't like blood, I won't go into it any further than that. But and she could stitch them up, do some other things, you know, surgically, and then they'd go through a rehabilitation process, and they'd be released. I guess I can stop sharing. There we go. So that was amazing, and that really encouraged me to shift my interest from specifically just research to more of a clinical medicine side, because I wanted to do more hands-on work in addition to the research applications. And I'd say for any of you who are potentially considering either route, you're confused, you don't even know what options are available. I certainly didn't at the time. You're able to do a lot. So now I'm a third year in the program. It's a 4-year program for veterinary medicine, similar to human medicine. And you learn mostly dog, cat, horse, cow. Really nothing in the curriculum relates to aquatic animals. There are a few classes depending on what school you go to. I'm fortunate that one of my professors, who is fantastic and might still be on this call, teaches a class about fish. So I got to learn a little bit about fish medicine, but it's a lot of external things. So I took a month-long course last summer called Aquavet, which is an intensive 1 month long of here are all the experts in the world on pick an aquatic species, say the word medicine, there you go. And it's fantastic. You learn so much, but it's not necessarily part of your curriculum. So if you are intending or thinking about going down this path, feel free to reach out whenever. I'm more than happy to chat with you or connect you to other people depending on your specific interests. And B, it's a lot of you taking the initiative to learn things and doing things outside. So the research that I'm currently working on is not at all related to my schoolwork, but I still want to do marine mammal behavior stuff. So I've been working on the side with that. And then you do a lot of externships, which is the like veterinary term for like what you would call a college internship type thing. So you can go spend time at different facilities, figuring out what you like. So I'll be going to a couple aquariums and other facilities next year where I'll be working directly with the veterinarians there and getting to learn that clinical side for aquatic species. And then if research is your thing and you're like, but I love this and I want to go do field work and stuff, the nice thing is you can actually do both. You're really able to do almost anything. And that's what I've learned by hosting Aquadocs, the podcast, which if you're a student, I would highly recommend. We do a lot of great conversations about what opportunities are out there. Every time I speak to a new guest, I'm amazed at whatever their job is. I didn't know it existed beforehand. And then I'm like, that's the coolest thing ever. I definitely want to go do that with my life. And a lot of people are making their jobs. It didn't exist until they had it. So it is a difficult field, but I would encourage you that, with anything relating marine mammals, sometimes it's hard to get into, but if it's what you care about, there are ways to make it possible. And it's such a beautiful, supportive community that even as a student, right? Like Teresa and I, congrats to Dr. Eric over there who just defended his dissertation, but Teresa and I are still students and the society is allowing us to have this opportunity. So it's a wonderful, supportive network of people. And, I could talk all day about it, but I will stop there and just say that if there's anyone listening, or if you know of students who are interested, I certainly don't have all of the answers, but I'd be happy to talk with you or can help direct you to other people who could help you out.
Perfect. Thank you so much, Michelle. That was fascinating. Really well presented. Obviously, people loved it. You had lots of great questions that came in. For those who are watching, the reminder that the recording is available on Facebook and is also going to be posted to the Society for Marine Mammalogy's YouTube page, and that the paper that all this research is summarized in, or published in, I should say, is available open access on the Marine Mammal Science website. So if you want any of those references that Michelle was talking about, or get the nitty-gritty details, that's a great place to look. And then, of course, as she said, reach out to her. And so with that, congratulations, Michelle. Like I said, fantastic, fascinating. We're going to go ahead and wrap up. But yeah, thank you everyone out there for watching. And hopefully we will see you in the new year. TBD, we have a couple things lined up, some pretty exciting talks coming up. So keep an eye out and we look forward to talking to you all soon.